So in this video series, I'm going to be going over some IB questions from topic 3.5. Specifically, it will be paper 1 questions, so multiple choice questions. I'll be making many more parts to this, so just stay tuned and keep watching them before your test. Now, I won't be going into too much detail in explaining why the answer is the answer. I'll be going into that briefly, but if you really want to understand the concepts, you have to check out the videos I made regarding 3.5 that I put in the description. So hopefully you watch those before you do these questions. Okay, so every time I'll open up this thing so you can try it out by yourself, and then I'll explain it. So give it a go by yourself. So the question is, how is the polymerase chain reaction, or PCR, used? So the PCR, I'll explain briefly, is a process that you use to turn DNA, like little DNA. So for example, here we have a cell. Say we only have one cell, and we know it has DNA inside. And what we want to do is we want to duplicate that DNA many times until we have millions of copies. So what they do is they remove the DNA from that cell and then put it through a process called PCR. So this blue arrow here represents PCR. And then after this process, you would have been able to make millions of copies of DNA. And the purpose of this is to be able to have sufficient amount of DNA to be able to do certain testing, to be able to determine who perhaps the murderer is and all that kind of thing. There's many uses to this. So that's the basic process. So let's see which answer matches this one the most sufficiently. So A says to make many copies of a DNA molecule. So far that sounds like what I just explained. That sounds correct. But we have to eliminate or say why the rest are wrong in order to be sure. So B is saying to cut DNA at specific sequences. So no, PCR doesn't do that. What actually does that is something called a restriction enzyme. A restriction enzyme, if you watch my video, you would know it's kind of like a scissor, and it cuts DNA at specific places so that you can do certain testing later. So if you don't understand this, you need to check out that video. So C, to splice fragments of DNA together into a plasmid. So this is also not true. This technique is just, this involves something called gene transfer. So it's not involving PCR at all. And the last one is saying to separate fragmented pieces of DNA based on their charge and size. So no, what this sentence is describing is something called gel electrophoresis. This is normally the process you do after you do PCR, because normally in order to do this process, this gel electrophoresis process, you need a lot of DNA, and so first you need to do PCR. So this is a different process. So the answer is definitely A. Okay, let's move on to the next one. Okay, which technique causes fragments of DNA to move in an electric field? So give it a go yourself. So let's go over the answers. So A, polymerase chain reaction. So we just did a question on that one, and we saw PCR is what makes a lot of DNA. So it's not involving any kind of electric field. So A can't be the answer. B, genetic modification. No, genetic modification does not involve an electric field either. This is completely a different topic. This is when you change certain genes of a specific species in order to make them better. For example, you can change certain DNA about cows to make them have better meat or something like that. This doesn't involve anything with electric field. C, therapeutic cloning. So no, therapeutic cloning is when you copy an organism and paste it, basically. You're duplicating an organism. So this doesn't involve electric field either. Gel electrophoresis. Okay, so this has to be the answer if A, B, and C are incorrect, but let's make sure it is. So gel electrophoresis, for those who know it but are not 100% sure, this is basically, I explained this in my video, this is basically when you have pieces of DNA from different individuals. So say we have a bunch of pieces of Ronaldo's DNA, and we put them in this machine. This is a gel electrophoresis machine. DNA is negatively charged, and if you've learned before that negative and positive charges attract, so opposite charges attract, and so when you put the DNA in this slit, this little slit here that the arrow is pointing to. Once you turn on this machine, that means you're turning on this positive charge. Then the negatively charged DNA will travel towards this positive charge. And depending on the weight of this molecule, so if it's lighter, you can imagine pulling a lighter thing is easier than pulling a heavier thing, right? So the lighter pieces will travel the furthest. They will move from all the way here, the furthest, to this positively charged electrode. And the heavier pieces, for example this one, don't travel at all because they're so heavy. So this is what gel electrophoresis is. And you can see it involves DNA moving in an electric field. This is an electric field. Something that has negative and positive charges everywhere, that's an electric field. So this would have to be the answer. Again, if you don't really understand everything, you need to check out the video for the full explanation. This is very brief. So let's go on for one more in this video. So this one's tricky. What is produced by somatic cell nuclear transfer? So if you have no idea what this even is, you might not. This is a process that is used in cloning. This is the name of the process that produces a clone. So first try the question by yourself. Okay, now I'll go into explaining what specifically this is, and then we'll check out the options. So a somatic cell nuclear transfer is the entire process of cloning, and the name makes sense if you understand what cloning is. So cloning is when you take, for example, say we want to clone this guy. Say we want to clone Sean the sheep. What the scientists do is, in order to clone something, you need its DNA, because the DNA is pretty much the instruction manuals to how he looks the way he does. So what they do is they remove the DNA, okay, from that somatic cell. This is a somatic cell. A somatic cell is a body cell. It's basically any cell in your body that isn't a sex cell. So any cell that isn't a sperm or an egg cell. And they use this because they want its DNA. If they have its DNA, now all they need is an egg cell. Because an egg cell, you can think about it, when reproduction happens, it happens in the egg cell, right? So the egg cell is going to grow and duplicate and become you eventually. But they don't want random DNA. They want specifically Sean the sheep's DNA because they want to replicate him. So after removing the DNA from his cell and removing the DNA from the egg cell, they're going to put his DNA into that egg cell, because now this egg cell will grow and become him. Okay, so after it will duplicate, and like this right here, after an egg cell duplicates, that's called an embryo. So you're basically forming an embryo with specifically DNA from Sean the sheep, because after it splits and grows up after a long period of time, it will become Sean the sheep. Okay, so how does this make sense according to the name? So somatic cell nuclear transfer. So you're transferring the nucleus from a somatic cell, right? We transferred the nucleus from this somatic cell into an egg cell so that this egg cell can become Sean the sheep, okay? That's basically what somatic cell nuclear transfer is. So do you make adult sheeps from somatic cell nuclear transfer? No, you don't. You make embryos. I just showed you make embryos, and these embryos may eventually become adult sheep, but you don't directly make adult sheep by somatic cell nuclear transfer. You make an embryo, okay? So technically you can after a while if it grows, but you don't directly make adult sheep, so it can't be A. So B, cloned embryos. Yes, that's true. You do make cloned embryos. That's what I just said. Because these embryos will grow just like you did in your mom's uterus. It will grow over time, over 9 months or whatever amount of time it requires, and it will turn into a baby, which will eventually turn into you into an adult after many years. C, so we know B is the answer, but we're going to have to eliminate C and D just to make sure. So C is saying rooted stem cuttings. If you don't know what this is when you have you ever tried that, to cut a plant and then replant it somewhere else to see if it can grow again? Yeah, that's basically what rooted stem cuttings is. D, genetically modified food. So no, to make genetically modified food, you don't use this technique at all. You actually change the DNA of the of the plant that makes the food so that it can make better food. That's genetically modified food. You don't use this technique at all. So I hope these question videos help you out, because teachers tend to always use these in tests because they don't make their own questions. They get them from the IB. So if you do enough of these questions before your tests, you should notice a lot of them repeat, and actually these exact same questions might show up on your tests. So it's very useful to do, in my opinion. So I'll make part 2 and 3 after this, and I hope you found this useful.